Thank you very much. And I'd like to take the opportunity to welcome everyone to the Single Cell Spatial Analysis Program Kickoff event. In this symposium, we'll talk very briefly about the highlights of what single cell and spatial analysis offer, and I'll introduce the Single Cell Spatial Analysis Program. Then we'll have a session led by Tom Wilson on describing single cell analysis and spatial analysis resources at the University of Michigan. That will be followed by our keynote speaker, Dr. Sumin Lee, to profile an in-depth approach using this type of technology. And finally, Jun Lee will introduce several new methods being worked on at the University of Michigan. First, starting off with the basic concept, so we're on the same page. What is the challenges in cell biology and medicine? Well, one is identifying cell populations. When we look at very complex tissues, such as brain, or different functioning areas, such as heart, it can be a real challenge to look at subpopulations. One of particular interest is, to me, are tumors, where we can really look at homogeneous looking tumors. However, if we look under the microscope, again, the tissues can look very similar, but using special stains, we can identify various subpopulations or very rare cells. This is exemplified here in this very simple model of a 3-cell tissue. If we were to take this tissue and grind it up as the standard method is in a bulk type of approach and look at the RNA, we'd get one value of RNA, for example, a value of 4. This could represent a distribution like this for each of the individual cells, or it could represent a totally different distribution of this. Presumably, these would have very different biological implications in their function and what these cell populations can do. So getting that information can be very important. Again, this bulk analysis will really give us an average gene expression from the whole tissue per cell or from all the cells together. If we can divide up those cells and get the information from each cell type, we'll start getting how each cell type varies and get a greater appreciation for the heterogeneity of the tissue. Because of the power of the single cell sequencing based methods, it was selected as Method of the Year by Nature Methods in 2013. And indeed, not just single cell transcriptomics, but many other single cell analytic measures can really help inform about the biology of disease and medicine and biology. And these include looking at the genome, transcriptome, cell mechanics and biophysics, the proteome, cell matrix interactions, the secretome, and intracellular stimulation are all important events that can be appreciated at the single cell level. Applications of single cell technology have really grown in the past few years and include developing human cell atlases for different tissues, rare or novel cell identification, drug-resistant clones or minimum residual disease identification. It'll have applications biopsy diagnosis and prognosis, and identify cell lineages in terms of biology and mechanism. These very important areas are now being expanded on by single-cell multiomics. We're starting to realize that we can look at proteins and RNAs at the same time, or DNAs and proteins, and even look at epigenomic type questions with proteins and mRNA. Because of the power of being able to do this multiomics, again, single-cell multimodal omics analysis was selected as Method of the Year in 2019. Even with the power of the single cell applications, we can generate incredibly high molecular content for single cells. A drawback to this is that we have lost our spatial context of the tissues. On the other hand, we can take a look at tissues and we have very high resolution imaging that can bring into focus incredible detail even at the submolecular or subcellular level. However, what we're hoping for is to merge these two together and get really good in-depth spatial analytics where we can see high content with spatial relationships, and the fields are starting to get there. This would have important applications in biology and medicine in a variety of different areas. Examples as we move forward is we're hoping to be able to take this single cell transcriptomics, be able to do spatial transcriptomics, and using the information, we can deconvolute those data so that we can really get appreciation of this high content at the single cell level within a spatial context. And with advances in artificial intelligence and bioinformatics and machine learning, we're being able to get in-depth approaches and gains to the biology and medicine of multiple diseases. We can get cell subtype special relationships, signaling network location, and patterns and relations to clinical outcomes. Now, there's a variety of spatial analytic methods now in existence, and they are expanding rapidly. And I'm not going to go through all the details here, and you'll hear about the ones at University of Michigan in the later afternoon session. But they're up and coming, some are in place, and some are being developed, and they're very exciting advances. The opportunities that spatial analytics can afford us are really only limited by one's imagination. Of course, it will provide more information in this novel context of space, allows multiple potential biomarkers in a single slide. In the past, this could have needed multiple sections, or we would have lost that spatial context. This is very important, particularly for rare samples that can be a challenge. We can co-localize proteins, RNAs, DNAs, and cells. And finally, a challenge that's really coming up and an opportunity is the need to develop new data analytic methodologies. We're getting terabytes of data from even one individual experiment, and being able to handle this data and the amount of samples tissues is a new frontier. A couple of examples just to kind of highlight some of the opportunities that it can afford. One, we can look at density of cells. For example, in this particular paper, looking at primary versus recurrent head and neck tumors, if you look at these pink cells, which are T regulatory cells, it can be shown that in recurrent tumor, there's a higher density of T regulatory cells. Of course, that can have mechanistic and prognostic implications. Again, we're not using these for just anatomical spaces and spatial relationships, but actually being able to understand functional differences. For example, here's one showing how multispectral imaging, which is an antibody-based approach, can determine immune cell density along a melanoma margin. And here we'd have a margin of the melanoma itself, and actually taking measurements of distances represented here by these lines can show indications of how that may lead to potential therapeutic and or diagnostic outcomes. An example is imaging mass cytometry-based spatial analysis can be used to predict lymphoma outcomes. Or we can get spatial temporal atlases of development, in this case, human intestine. Over time, we can see how it changes, how the cells change, again, within the context and the actual spatial activity. Finally, spatial transcriptomics can identify pre-invasive cells. So perhaps a prognostic indicator, knowing whether this may inform clinical care. Because of the power, again, of spatial transcriptomics, that was selected a method of the year in 2020. All of this activity has been ramping up at an incredible pace. And we wanted to take advantage of that because we know this technology, particularly the single cell analysis and spatial analysis, has been amplified within the university. And we wanted to bring together the resources to be able to help that and help our investigators at the University of Michigan. So a team of us put together the single cell spatial analysis program, where we're really focused on getting that high molecular content within the high resolution imaging into focus and being able to afford opportunities. Our vision is to enhance the University of Michigan's ability to apply spatial analysis to drive next generation solutions in biology and medicine. And our team leadership is very multidisciplinary. We have application experts, bioinformatics experts, bioengineering, genetic experts, and we come from a variety of different departments and several different units from the university. Our program goals that we're moving forward on is faculty recruitment. We have 3 slots that we're going to recruit faculty. We're looking for trailblazers in the spatial analysis field to be able to bring those people in and help move Michigan forward. We're going to enhance research resources. We've already done that. We've purchased some incredible instrumentation, NanoString GeoMix, which allows for spatial analysis. We bought DNA single cell sequencing application, the Mission Bio Tapestry, and then put that in the advanced genomics course and the single cell course. And of course, community building. We're going to be establishing monthly seminars and workshops. We'll be having pilot projects. So stay towards the end to hear a little bit about that. This is what we envision our success. We're going to have a cadre of investigators at the University of Michigan focused on these problems, working together. We hope to develop discoveries that'll be high-level discoveries at the university and throughout, and finally enhance funding opportunities by making those resources available to our investigators. So of course, we need to thank all of us support that has enabled us to put this program together. This was funded by the Biosciences Initiative, and other partners have helped provide funding for equipment and pilot projects, including the Rogel Cancer Center, the Forbes Institute for Cancer Discovery, the Biointerfaces Institute, and the Office of Research at the Medical School. So I'd like to end there and thank everybody that's participating, thank all our speakers ahead of time, and hope that we all have a great conference. And now I'll hand this over to Tom Wilson. Thank you very much.